Hello and welcome to the Deep Sea Podcast, a punk take on a science podcast about everything deep sea. I'm Dr. Thomas Lindley, Curator of Fishes at the National Museum of New Zealand, Te Papa Tongarewa, and with me is the Professor Alan Jameson, OBE and Don't You Forget It, Director of the Deep Sea Research Centre at UWA in Australia, and Lead Scientist for Inkfish. Hello, mate.
Good evening.
Good evening. Happy Sunday.
Yeah, it is Sunday. We're still working. That's not fair.
We're recording this a little bit earlier than we usually would because I am going away into the wilderness and I don't think I'm going to have any connection. So we're trying to get this one in the bag a little bit in advance. So if they find Megalodon or drain the deep sea or something like that happens and it's not in the news section, sorry. We're a little earlier than usual.
Actually, we're not. We're actually on time for probably the first time in 59 months.
Yeah, that's probably a better way of thinking about it. So I'm off to Norfolk Island. It's in the Australian territory. It's just north of New Zealand, between New Zealand and Australia, teaming up with Sydney and Auckland museums to characterize the unique life in the region, which apparently includes lots and lots of sharks, and we're diving for this one. So this isn't deep sea at all. This is part of my new wider remit. So I have to dive and get fish with sharks that might be enticed by said fish.
Do you have to collect sharks by hand whilst diving?
Sharks may collect my hand, is how it's going. We're going for the little cryptic things.
Right. You're not just going to wrestle like a 6-meter shark, try and bring it up by the water.
Well, that's the worrying bit because it means I'm going to be all sort of up close and looking at the coral and not noticing the tiger shark that's showing an interest from behind.
Good luck with that. That sounds great. Well
so
done.
if this is the last episode, avenge me.
Enjoy yourself.
I'm not going to be all noble. You know those people who get attacked by a shark and go all noble and they're just like, oh no, they're a real advocate for sharks. If one attacks me, no, avenge me. Just pour bleach into the ocean.
Yeah. It's like other people in Australia keep telling you that shark attacks don't happen that often. They do, right? They happen way more often than
it's the tourist board hiding all that. What have you been up to?
Well, I've been up to not very much.
Oh,
I have been up to quite a lot of things, but not much I can talk about.
Okay.
Just trying to work up lots of bit and publish lots of papers and organise everyone's life. It's lonely at the top, Tom, when you're the director. I'm the only one here at the weekends.
Recording a podcast for no money. But for good.
Exactly.
But for good.
I guess so. But the first time in a long time that I've done 2 consecutive episodes from the same country.
It is. You usually move around a lot more. Just want to say a huge thank you to our patrons who continue to support us and make the show happen. This week, we would like to thank Rainstorm Wolf, Shia, and Racist Teacup.
All right, okay, I've got some questions here.
I didn't hear back from Racist Teacup to get confirmation that it'd like to be called Racist Teacup.
I'm assuming Racist Teacup is not this person's real name, but I'm going to question Rainstorm Wolf. If that's their real name, that's the most heavy metal name I've ever heard.
They've both surfaced on the Discord, which is our little online community. And if you're on the fence about joining the Patreon, it does get you access to that. And there's some good, lovely chats going on out there. Actually, stalwart and linchpin of the Discord, Megan has taken on helping out with the podcast a little bit. So she's gone from fan to supporter to on the inside. So she's helping out with gathering some of the stories and doing the graphics for the episodes. Do you have a song of the month?
No.
Oh.
Make it Ozzy Osbourne Crazy Train because my son just learned how to play on the guitar and it's brilliant.
There we go.
Well, I can send you the video of William playing it and just play him. Is that still copyright?
Oh, that's much better. Yes, please.
All right, here's my son William playing Crazy Train.
Nice.
He listens to this show as well.
Does he?
Yeah.
Hello, William. It's been a while.
Yeah. He's also now making money out of his YouTube channel. 14 years old.
How did he do that? Because we aren't.
Because he's got a significant following. Yeah. He's makes train videos about commuting trains.
Wow.
There you go.
Good on him.
He's famous in his own little social circle. Profiteering off of the internets.
Can we hire him?
I think we probably should.
I know. So to jump into some news, recent paper came out basically trying to quantify how much of the deep sea we've actually got our eyeballs on. We actually have visual data for, calculating it at 0.001% of the deep sea has been seen in the last 70 years was the sort of catch-all at the end. Similar to the moon analogy, that is a real sort of generalization, worth a little bit of discussion about what data was included in that, what counts as seen, and if that is the best way for us to be doing science. And I know we had loads of chats on the Discord about this. I haven't had a chance to properly catch up with that.
Well, there's chats about this thing all over the place because I think it raises more interesting discussion points than it does. It's an interesting paper. I don't want to slam someone else's paper, but it's fallen victim to a lot of the stuff we talk about. If you read that paper, what it says is we're doing more deep sea research than ever. In the last 50 years, it's increasing significantly. The number of institutes, the number of dives, you could do the same thing on a number of there's a massive upward trend. And so there's all these successes in there. And then it just leaves you with, and it's all futile. And so it feels like the headline is what this paper serves to try and defend. But you could write this positively or negatively. And that 0.001% is not going to go away anytime soon. That's here to stay. There's people from my lab and from Heather's lab in Florida just now at a conference, and apparently that phrase is already finding its way into any deep sea talk, because it's almost like we need another headline to justify what we're doing. And I think the danger is if I was a funder and I was like, what am I going to put my money into? And you think, 70 years, you've done so little based on this thing that it's probably not worth putting any money into it because it's futile. And it doesn't take into consideration that the definition of exploration to me is very troublesome. And that's because I've worked a lot with philanthropic people. So we've done loads of dives and all the rest of it. But I would say many of those are not exploration, right? They're confusing adventurism with exploration and things like that. We did 22 dives, I think, to Challenger Deep, and not one had a scientist in it, and not one was ever reported scientifically, because the point of going there was just to be the deepest X, Y, and Z, right? So they count as 22 dives for exploration, but we didn't learn a single thing. I was chief science of that project, and I don't think I've even got a copy of the videos because they were driving so fast and so far off the bottom. It wasn't, the exploration has to be about the place. Adventurism is about the person. So there's a lot of dives that have taken place which are centered around the person, not the place. And then you get into science, and science is not the person or the place. Science is the question. Now, these phrases like 0.001%, the seafloor, all the rest of it, is ignoring the fact that we are very good at statistical sampling and design. We don't have to photograph every square inch. It depends on what the question is. If the question requires you to see every square inch, then that's fine. But why would we want a picture of everything? You see, I mean, it's like self-flagellating for a problem you never actually really had. And yes, it would be great to do more deep sea research. Of course it would be. But I think these types of sound bites are probably more harmful than they are good. And it'll become another rhetoric that you hear every single time someone wants to justify why they're doing a deep sea thing.
Yeah. It'll be the new moon analogy. It'll be the new intro. It'll be the start to every pop culture thing on the deep sea. It seems staggeringly low, but anything where you can't just fly a drone over it. So anything like with a canopy, I'd say the native British woodland would be less than 1%. Has ever been seen.
Yeah, how much of the forest floor have we seen of the Amazon, or how much we've photographed, not how much we've seen, how much we've actually imaged? Probably not very much. And all these analogies are not fair. My point I'm trying to get across is like, yes, there's a number that's been calculated. I think it's only captured a very, very small amount of effort. I mean, the fact that the UK, for example, doesn't even come up in the heat map kind of thing is, and the mere submersibles don't appear to be there or whatever. I don't know. That's not the point. The point is why try to come out with that statement? Or is that to try and encourage people to fund it? Or is it to say that we haven't done a very good job? That negative headline feeds into the conversations. I mean, I've Googled a deep sea on the news page, and the way that's been reported already is just terrifying. It's just like, here we go. Not one of them says, the number of countries, the number of institutes, and the number of dives has increased significantly in the past 30 years to do with deep sea exploration. Those are the little sound bites.
That exponential graph.
Yeah, they will just not get reported because we as a community, if you want, it's given them this killer statement that the deep sea is still a big mystery. There was one particular one that was, let me find it. The headline is, over 99% of the deep ocean seafloor remains a mystery, a study finds. Now, it doesn't, does it? It's not a mystery at all. Like, the abyssal plains are big of a planet. We know what the abyssal plains look like. We don't have to see every square inch of it. There might be some cool stuff out there that we're kind of overlooked, but generally speaking, I don't remember ever getting in a subway and reaching the sea floor and being absolutely, completely 100% bowled over, like, mystery solved.
It's all jelly cake and ice cream down here.
Yeah, you know what I mean? And it's playing this, I don't know. It's a difficult one because someone's always spent a lot of time into it, but I think this is one that's going to go round and round and round in circles. And I don't know what the point of it is.
The sort of point was to render it down into a single factoid that's sort of press ready and introduction to funding application ready and PopSci article ready, but
Yeah.
flipped the wrong way, really. It's a little bit negative. And I think people might interpret that in lots of different ways. I mean, we've said ourselves, one of the things we've said a few times is, more eyes in the water, more eyes in the deep sea. And that's how we're going to see things like these rare squid and, things you can't plan for. But that's excited, forward-looking, yay, let's do this attitude rather than like, oh, why bother?
Yeah. And there's also that, this is not just this paper, there's other papers out there as well that have talked about this bias towards certain countries. And this in particular, when it talks about all the effort has been biased towards America, Japan, New Zealand, France, and Germany. But it's whenever this comes up in some of these opinion articles and stuff like that, that's almost presented as a negative, as if to say that these countries are not taking away opportunity from anyone else. It's just they have an interest and a means and the money. They see value in deep sea research, right? There's many countries that don't care, or they don't have the capacity to do it, and it's an inherently mega expensive thing to do. And it's written as if these countries are doing something wrong. But what I read into that as well, congratulations, America, Japan, New Zealand, France, and Germany, you've nailed it. You're doing really well, right? And let's get everyone else behind you and get involved. But instead, it almost feels like, well, they have all the toys and they're not playing. Maybe that's just been paranoid because there's all these ridiculous parachute science papers out there that it's like one country spent a lot of time and money and effort trying to do something for the greater good of human knowledge. And it's like, well, you didn't involve enough people, therefore. And we should be encouraged and celebrated when people do a lot of cool stuff, and just understand that just because one country does it doesn't mean to say that every country does it, or just because we value something doesn't mean to say that we have to assume that everyone else values it too.
Above other much more pressing
There's
things.
no balance to the argument. This is my point. There's no balance to it. It's weird seeing something as subjective as where it's not a balanced argument. I don't think it's agenda driven.
And there's definitely data missing.
Oh, hell yeah.
It depends who is open with their data.
Yeah, but there's other things as well. There's a thing in there which is an interesting aspect of early exploration. Is a map showed, I think it was the 60s or whatever it was, that people used to just go sample all over the world, and then recently there's been a trend towards within your own exclusive economic zone. It's like that's because in the 60s there were no exclusive economic zones. Once you left the shore, there was no imaginary line there, right? So people went around the world to discover things, and they did. But the whole point of going really long distances and everything else is everything the same? Look at the sea surface temperature. We've eventually got satellites, all these other things. We did all that. But now, deep sea research is so inherently expensive, and so is fuel costs for ships. You've got to have a pretty good argument for taking a ship on a 35,000 nautical mile journey. Plus, a lot of these institutes and resources are funded through the government with taxpayers' money. And now, after 1984, because of the fishing dispute between Peru and Chile, they came up with a magic number, which was 200 nautical miles, drew a border on it. So now governments and taxpayers are like, well, that's where we want to see our money invested.
Yeah.
Because
why are we paying to research someone else's?
Yeah. You're not going to get a ship in New Zealand and go, right, we're going to go all the way to Japan and see what they've got in their waters. It's like, no, taxpayers are like, wait, we want to keep tabs on what we're doing because that's what we pay tax for. And so the ocean used to be for everyone, now it's not. And the annoying thing is the second E, an exclusive economic zone, means economic. And there's no economics in science. So there's no reason for the EEZ really to be interfering with scientific exploration. And if you read the declaration, it actually says it doesn't. You can't stop scientific investigation. But these imaginary lines are such, then human nature is such that when you draw them, you put a fence around yourself. It's just easier to work within it. And so the argument's much more complex, I think, than just people are being selfish and only work off their coast. It's like, one, of course, they do. Two, it's the most efficient use of fuel. Three, and it's what I'd imagine taxpayers are moving towards.
In more good news, bottom trawling stops the ocean from absorbing carbon dioxide. A new study from GeoMar highlights the impact that bottom trawling has on the ocean's ability to absorb CO2. During bottom trawling, the sediments on the seafloor are resuspended into the water column and significantly increase the release of carbon dioxide into the atmosphere. So disturbing the seabed with bottom trawling releases not only organic carbon into the water, but also intensifies the oxidation of pyrite, apparently, both of which are especially evident in the fine-grained muddy sediments of the deep ocean. So for those unfamiliar, pyrite is a mineral typically found in oxygen-poor seafloor. When it's agitated into the ocean, it reacts with oxygen, generating acid that converts climate-neutral biocarbate into the gas carbon dioxide. Oh, there's some chemistry going on there. And apparently the similar things happen when you till fields on land. Trawling's often been compared to ploughing a field. Apparently you see the same thing there. There's art. There's art in the Mariana Trench.
Oh, has someone done an art?
Someone's done an art. And I was worried at first, but I'm okay because the art is attached to something else and it's not just being thrown in there.
Rakshu Momambabu is an artist, architect, and fashion designer from Singapore. There you go. Carry on, Tom. I've done the name for you.
Thank you. You've done the reading bit. Oh, now there's more reading. Has collaborated with New Star Technologies and Jamstech, I know them, I remember them, to place an art installation on the ocean floor. Laksham's latest work, a set of 3 hollow metal cubes, each featuring an interlocking arrow-cut design, are 10 centimeters or 4 inches square, and they're now located 7,000 meters, 23,000 feet below the ocean in the Mariana Trench. 2 of the cubes were made of a special corrosion-resistant stainless steel, and the third was created from 80 layers of ultra-thin stainless steel fused to 4mm thick wall. The cubes are now part of Jamstech's long-term borehole monitoring system, or LTBMS, if you want to be catchy. And the monitoring systems each have a special cavity to house the cubes. The LBTMS was originally deployed to monitor seismic activity near tectonic plate boundaries, which is a high priority for Japan. But at just 10 centimeters long, if you drop that right into the axis of the trench, that's not going to hang around for long.
I think it'll just go straight into the mud.
Yeah. It's really soupy at the bottom.
Yeah, it sounds like it's one of the worst places in the world for super soft mud. It's like porridge.
And amphipods dancing in and out of it.
Polychaetes too.
Do they?
Scale worms. Yeah. Still that a bit in Tonga Trench as well. Yeah, they love the old 10,000 metres.
Deep-sea fish have independently evolved the same gene mutation for pressure. So in a new study, scientists have taken a look at the genetics of 11 species of deep-sea fish living at depths between 1,218 and 7,730 meters, or almost 4,000 feet to just over 25,000 feet. Genomic analysis showed that deep-sea fish have a lower mutation rate but strong signals for natural selection, especially in genes related to DNA repair and membrane integrity. Most fascinating of all was a mutation in a gene called RTF1 that was seen in nearly all fish that live below 3,000 meters, so below about 10,000 feet. According to the study, this is a clear example of rapid convergent evolution, and wildly different species separated by millions of years all arriving to the same genetic solution to survive in that high-pressure environment. Through this genetic analysis, the researchers confirmed a centuries-old theory that deep-sea fish didn't follow just one path into the abyss, but 2. Some are ancient survivors, pioneers that braved the depths before dinosaurs vanished, and others are new immigrants, latecomers that emerged after the mass extinction 60 million years ago. What is it Torben Wolf called these 2 groups? It was him that first came up with this, wasn't it?
No, I think it was the Russians. I think it was the guy Andreashev.
Oh, was it?
I think I remember that was the first time I came across.
Andreashev of snailfish fame.
Yeah, he was talking about or could have been birthday. I don't know. There was a whole bunch of old Soviet guys on the Vichaz and whatnot. They refer to this as the ancient invasion and then the secondary invasion. That's pretty cool that they were right. And that thing was written in the 50s.
Yeah, purely based on morphological observations, because some groups had whole novel structures, like an anglerfish, the lure, the body shape, lots has changed about that.
Yeah.
So that needed a lot of time. Whereas things like the snailfish, they look, all their adaptations are biochemical, really. They look not far from their shallow water counterparts. So they've rocked up late.
Yeah.
And it's interesting about what may have offered that release, what's allowed these to get in there now. And I do feel like most of the old deep ones are pelagic. The more recent comers, this is just a hunch, but I feel like the more recent comers are the ones that tend to be on the bottom, the really deep ones.
Yeah.
Nah, fair enough. But speaking of genetics, that leads us on to our interview.
Yes. Complicated genetics based around a small glass of water.
Magic.
Evil
magic.
Yeah, the guest today is some sort of scientific sorceress who does magic science with water. That's how I would explain it.
I mean, that's pretty much it.
Yeah.
No, I tell you what it is. It's homeopathy.
Yeah, basically.
Water has a memory, and she can take a tiny bit of memory out the water.
Yeah, that's it. It's the souls of the animals that are no longer with us have been preserved, and she managed to conjure them back to life into an enormous spreadsheet.
Magical.
Yeah, but like a sorceress bit on Excel.
Oh, DNA homeopathy.
Yep.
Okay, let's talk to Georgia.
We're talking eDNA. So joining us today from Old Mate's office next door is Dr. Georgia Nestor, who's going to talk about eDNA. So let's kick off with what exactly do we mean by eDNA and how do you do it?
Thank you for having me. So environmental DNA just refers to any genetic material that's left behind in the environment by organisms. So we can capture that genetic material, that DNA, and then see what species were in the environment at the time that we took a sample. So because it degrades over time, it sort of provides like a here and now view of what was in the ecosystem at the time that we took the sample.
How long is the DNA detectable in the environment after the animal is left?
So it depends really heavily on the environment that you sample. So if you were in a really nice cold environment, the DNA is preserved a little bit longer. If you're in like a really tropical, hot environment, it degrades a lot quicker. But modeling has been done. We've sort of seen that it's about anywhere between a few hours to a day max.
So is it easier then to do it deep sea than it is to do it coastal and shallow water because of the high pressure, low temperature thing?
Not really, because of the low biomass. So in the tropics, there's a lot of fish and there's a lot of species, there's a lot of corals, there's a lot of DNA. In the deep sea, however, it's a little bit more barren. So we're trying to just grab as much as we can out of that environment. So, we might see like a few fish in our videos, maybe 10 max, and I'm just struggling to amplify that DNA up as much as I can.
Oh, that was a very polite way of saying yes, but you're wrong.
I'm like, good idea, but no.
Yeah, absolutely not.
And what causes the breakdown? Is it microbial? Is it a delicious little snack? Or is it that it's such a fragile molecule to be knocking around without a cell to protect it?
Yeah, it's a combination of all of those things, really. So we, with environmental DNA, detect generally people are trying to move on to larger fragments, but really we're targeting those small degraded fragments because of how quickly they break down. Essentially, it'll be released as a whole cell, and then it just starts to degrade, where it be microbial, or it could be UV or temperature. All those things start to degrade the cell, and then we'll target those tiny little fragments that are left over. We generally target mitochondrial DNA because it lasts a bit longer as well.
So for a bit of context, I should probably explain that Georgia works at UWA and is one of the regulars on the ship that which we operate around the world. And so the eDNA research is a major thread in what we do. So now would be a good time to explain the mechanics of how we get the sample and what the sample is.
For our deep sea eDNA program, we generally use our lander systems. On each of our lander systems, we have a Niskin bottle. The Niskin bottle on the surface is rigged up with a galvanized link. So when it gets to the bottom after a certain amount of time, that link will degrade and then the bottle will snap shut. So we're hoping that the water that it captures is like only deep sea water. So it shouldn't be any of the surface water or the mid-water column. I also attach some sponges and filter papers to the side of the landers as a passive method of detecting eDNA.
What do we call them again? The puffs. So.
The poofs, we call them. Yeah, so the sponges we call the poofs just because it sounds fun. But they are polysyrethifer. I can never say the word, but they're a poly something sponge.
So this is one of these things that's interesting because there was a paper written not that long ago talking about how glass sponges are the perfect platform on which to study eDNA. Because it's a sponge, and that's exactly what we're doing. Do you have any data from those things yet to show that just cable tying a sponge to a lander is a solution? Because that's one of the greatest, cheapest sampling instrument attached to the most expensive thing ratio.
Yeah. So I guess when eDNA started, it was sort of like we can use it and we can do this water. And then I think over time, people are like, what if we just chuck a sponge on things? Or like, what if we, I was on a paper recently and it was like, what if we put a paint roller down a column? Will we see the same thing?
A paint roller?
Yeah, we were rolling pylons. It worked really well, actually, for benthic stuff. But with the sponges and the passives and the actives, because they work in such a different way, so the actives, we're actively pulling through 12 liters of water and we're capturing all that DNA on the top. I kind of was hoping that the poofs or the sponges would not work because they're a pain to do.
That's just what.
Yeah, that's just where I was like, hopefully this doesn't work. This is awful. Because we were pushing it through a syringe after each time. But interestingly, what I've seen so far from the Trans-Pacific stuff is each method captures a different portion of the community. So I was seeing a lot more of the diel migrators in the passive stuff because they're going down from the surface all the way to the bottom and then all the way back up.
So wouldn't the solution then be to do both?
Yes. So going forward, we're going to do it all. We're just going to keep doing everything. And if anything, I'll just attach more things just to see what happens because curious. And
so this is a bit of a loaded question because I know the answer to this, but this is the thing I find most interesting about having watched you and your colleagues do eDNA for hours and hours on end over the last 2 years, is that it's probably the least immediately rewarding way of sampling the BT.
Yes.
All you get is a bucket of water at the end and some little filter system that you got to freeze at the end. And we're all sitting next door with videos of sleeper sharks.
Yeah.
It's like, woo,
It's
look at our
it's
one species.
it's very interesting also to show people, like, the crew will come down and be like, oh wow, cool. So it always sounds so exciting. You get DNA from the water. What are you doing? And then they'll come and watch me filter water and they're like, see ya. That's awful. That's interesting, but no thanks. I'm gonna go look at the fish. But I think it is not very instantly rewarding, but the data that you get out of it is incredible. So I should probably explain that with eDNA, there's two routes you can sort of go down. One is the species specific route. We can look at like the genetic variability within a certain species and just target one taxa. Or you could use what I generally do in our deep sea stuff, which is metabarcoding, where you'll apply certain types of primers to amplify up as much DNA from as many taxa as you can, just to see what's there. So then afterwards, you guys will have however many fish you see on a video and whatever you see, and then I'll have 1 billion species to look through and see what makes sense and why they're there.
Yeah, that's what worries me. I remember you showing me the data from some of the previous trips, and it's just
Excel tables for days.
yeah, it's just insane. But so then we have getting the sample is actually the easiest bit in all this, I think. Then there's the boring bit of filtering it. Then there's oh my God, look how much data we have. But then there's a little problem, isn't there? Because we haven't got a reference library of every species in the sea. So then when these returns come in, they're only flagging to the nearest thing we have already in the global databases, right? So there's this issue of we might know how many species there are, but we don't necessarily know what they all are.
Mm-hmm.
So how do you deal with that?
The way that eDNA works, like you said, is we take our samples, we bring it back to the lab, we extract them, and then we'll amplify up the DNA through qPCR, and then we sequence those samples. Then it's a bit like forensics, where you'll compare it to a database of all known taxa at that time, or all sequence taxa, I should say. But like you said, there are huge gaps, especially for deep sea taxa, just because it's hard to get the tissue. When it comes back to the surface, it's generally, in a lot of cases, not the best quality to curate a whole genome, for example. So we might have only certain genes available for certain species. So what we do is I use a lowest common ancestor approach. So we might hit a Barythrides, for example, but we don't know what species it is. And I will pull that back to a genus level because only one of those Barythrides has a 16S gene available. So then I'll say, well, I can't confidently say which one it is because there's no reference material available. So I'll pull it back to a genus level. But the more that we sample and the more that we use tools like ROVs to actually pull up the taxa and barcode them, the better these reference libraries will get. And the good thing about eDNA is you can go back, kind of sounds like nightmare fuel, but you can go back and look at your data again and re-blast it years later and be like, oh, wow, look at how all of it's changed. Let me write another paper on that.
So does that mean you don't have to go to sea anymore? You just live off the current data set for the next 15 years?
I mean, you could, but we'd only ever find out about the Trans-Pacific. So...
Well, I think it's interesting that the one of the problems we have, deep sea, let's use deep sea fish, usually one of Tom's favourites, is the mighty Basidius is nothing short of an absolute pain in the backside because we know there are multiple species. We can see there are multiple species, but they're really cryptic because they all kind of look a little bit samey. And we only get to see the ones on the land, for example, that are interested in prey on amphipods that come to the bait. And we very rarely ever see them in the sub. And there's hardly any samples ever taken because they're just deeper than most people have ever trawled. So if we were to do eDNA alongside that, we assume we'll get lots of Basidius eDNA, right? Because they're right next to the bottle that's sampling it. But then is there a way of interpreting the data from your stuff to say how many species of Basidius are there, rather than just being there's a big Basidius soup in the middle?
Yeah. So I guess there's two approaches, and this is actually something that I am personally interested in using eDNA for in future research. One would be developing a species or genus-specific primer for Basidius and then trying to get that variation over and over again and using it across all of our samples. Because once we've extracted the DNA, we've got the DNA until we've run out, really. So we can keep analyzing it using these specific primers to see the genetic variability between those taxa groups, or species, I should say, and then see if we're detecting different species in different areas, and then hopefully do some sort of network analysis with that. That's one approach. Or something else that I'm super interested in doing is a taxonomy-free approach. So we get these things out of our sequencing called amplicon sequence variants, or ASVs. I want to look at, okay, we're in the Pacific, we keep seeing this one amplicon sequence variant over and over and over again. It's hitting Basisetus quite low. Then we move to Antarctic, we're getting a different amplicon sequence variant, but it's the same Basisetus hit. And then comparing those and saying, well, we've gotten these certain sequences over and over and over again. They likely represent these different species or variations of the same genus.
Yeah.
That's something that I'm super interested in. But it would take a lot of machine learning and AI and Bayesian models and time.
But this is one area of deep sea science which is absolutely moving into machine learning and AI now, isn't it? Because it's just your volume of data that comes in versus the volume of data you have to blast against. It's becoming a automation tool, right? And trying to figure out the best way of dealing with this, because going through these things manually, line by line, going, which is this really a myctophid or not, is not the most effective use of time. Plus, it would probably mentally destroy anyone who's trying to do it, which I'm sure you'll agree with.
Yeah, I mean, it's only made me stronger, but yes.
Has it though?
No, mentally I feel weaker.
Yeah, I've seen you slowly degrade over the last 2 years. Another big question for Eden, which is especially when you go into very deep water and you've got a period of time where you know that this fragment has left an animal. How do we know that a species detected in the sample is from that depth and not something that she's fallen down from above?
I think with pressure, it would be, if it had fallen a couple 100 meters, it'd probably be fine. But over such a huge gradient, it's quite unlikely that these small fragments aren't degrading beyond the point of us detecting them. So in some of my previous work, and I'm seeing it in some of the Trans-Pacific stuff, the things that really confuse the data are those dial migrators, the lanternfish, and all those ones that go, oh, I'm going to go to the surface now, and now I'm going to go to 1,500 meters, and I'm going to keep doing that over and over again.
Yeah.
They really mess with my data quite a lot. But with the Basisetus and the Barophrydes, I generally only get those in those bottom water Niskin samples, and then comparatively in the passive samples, I'll get such a small signal from those. In previous work that I've done in submarine canyons, I did get a lot of hits in the surface to, I think it was Basisetus, and I was very confused by that until, we had a chat the other day, and it was larvae. Makes more sense.
Yeah.
Because we were finding the larvae.
We should tell Tom about this, because Tom interviewed the people on the Blackwater diving episode.
Oh, it was such a cool one. Yep.
Yeah. I was showing Georgia that paper the other day, showing there's all these deep sea demersal. I mean, we've known about this for decades, but it's nice to actually see them.
Yeah, and I guess because this was my PhD, so I was a little baby, and I had a CTD, and I would do surface 100, 200, 500, 1,000, and then bottom water, and I was getting them in the bottom water, nothing. Surface, and I was like, oh, I've mislabeled these, or something has gone wrong. But then it makes so much sense after reading that paper.
Yeah.
And it's a thing we know, but I don't know, we sort of knew it without really embracing it. And now that we've got the Blackwater divers and people are like diving with Basisetus, it's really hitting home that, and the rat tails as well, they're all planktonic at the juvenile stage.
It's crazy, yeah.
So there's a little story I want to tell that is purely to wind up Tom.
Oh, fun.
And it ties in. The next question I was going to ask was, what's the most unexpected thing that this method has turned up? But don't answer that just yet, because I want to wind up Tom on this. So Tom just spent loads of time in Antarctica trying to find a colossal squid. Right? Very, very confident, he said. He said he was going to. He didn't say he was going to try. He said he was going to find the colossal squid. And he spent a great deal of time down there. And guess what? He didn't. Someone else, about 2 days after he left, did. Right? But then I remember when the first time you came into my office and said, if you get me water samples, I can do eDNA. I'm like, yeah, whatever. And you showed me the stuff you did off Australia. And one of the things that jumped out was, without even trying, you found the giant squid off Australia.
Yeah. I guess that's one of the super rewarding things about eDNA is when you look at the sequences after and you're Googling them and you're like, I'll get you hang on. And then you pop it in and you're like, yep, that's definitely the giant squid. And it's super rewarding. And it's nice because there's a lot of these species that they're monotypic. So it's not anything else. I'm hitting it at 100%. I'm getting these massive amounts of sequences. It's definitely there. And we're expanding species ranges and all those sorts of things. But I guess the downside is, you don't physically see it. So it's not as exciting. But I think
It's hard for the press release.
yeah, exactly. It's like, and we saw the giant squid in the DNA.
That's fine. This is just about getting one over on Tom, because we've just, for the benefit of the audience, so Georgia was on the same trip I was on in Antarctica. And so technically, in those samples that are currently in the freezer, we could have a little piece of colossal squid.
Mm-hmm.
That Tom
We had
doesn't have
all
that.
the primers, we had all the Niskin bottles, and a month and a half before we went out there, the permitting agency decided that they'd changed their mind, actually.
Really?
Yeah.
Really? For water?
Oh, and they wanted an ethics agreement because there might be animals in the water.
Nah.
Oh,
They-
well,
they-
they're not the smartest animals if they get those Niskin bottles.
They totally undercut us. So yeah, we took 2 full-size, paid a fortune in extra baggage to take 2 full-size Niskin bottles and all the primers in an ice pack, and it was a nightmare to get that across South America. And then they pulled it out from under us right there.
Wow.
That was annoying. Well, you wanted to wind me up, Alan, but you didn't know how much you were winding me up because we tried.
It makes it even better.
Yeah, I guess I find that very interesting because generally eDNA, that's one of its benefits is that, we're only going out, we're only getting water, we filter it, we put the water back into the ocean. It's just, we've taken the DNA away, but it's quite non-invasive in that sense. Like we're not extracting animals. I think the only time was when a Coryphaenoides or a rat tail got into the Niskin that one time, but that was a surprise. Apart from that, it's quite non-invasive. So I'm surprised, that you needed ethics.
I can honestly say there's been twice in my career that I've caught a fish using a Niskin ball. Yeah.
They're brilliant samples, though. They don't get much damage.
Yeah, well, those are some pressurized little, Niskins.
We had a snailfish in one off Atacama, which was perfect. And then I
Yeah.
think was it Tonga that there was a rat tail? It must have just been snuffling around.
Novocanton.
Was it Novocan? They're just snuffling
Yeah.
into Niskin, and then just as it fired, and we just basically grabbed it by the head.
It was so bad.
So unlucky, it's unreal.
But great for barcoding, great for making a whole genome.
Exactly.
Yeah.
His death was
Plenty
not
of
in vain.
DNA in that whole fish.
Yeah, think of the DNA.
I just fancied a bit of clarification because the wording sort of conjures this idea of loose, free DNA, like the actual molecule just loose and free in the seawater. Is that what you're mostly amplifying, or is it mostly individual cells that have been sloughed off?
Yeah, so it's mostly the free and extra because whole cells, I think that's sort of an avenue that a lot of people are trying to go down is getting those whole cells to look at actual individuals, but it just degrades and splits open so quickly. And then the pressure in the deep sea, even getting RNA is quite difficult. So we target those small open fragments, really, the small broken pieces.
Hanging around.
Yeah, pretty much. Like it's a big old soup of just split up cells, really. Whole cells would be great for looking at individuals, but I don't want to say impossible, but they've been working on it for about 7 years now. And in a fishery where there's lots of them, and it's still very difficult to get a whole cell.
And are you seeing differences in the high pressure adaptations? Does deep adapted DNA hang around for longer? And do you have more confidence that shallow water DNA would be broken down more quickly by the pressure as it fell?
I used to think that. I just sort of thought, oh, that makes sense logically. But then based on some of the work that we've done and the scales that we do it at, in coastal water, I've done things in the Perth River here and at really small scales. And within the space of like 50 meters, the communities are looking different. Like we're seeing a breakdown within spatial scales. But then in the deep sea, I thought, okay, well, it's just maybe going to be a bit of a soup and it's going to be quite hard for me to get these spatial patterns in the data. But I've seen the same thing in the submarine cannons I did off the coast of Australia or Western Australia here. We would travel, a kilometre or two, drop another CTD, and that site was different to the previous site, which I found quite interesting.
Yeah, because you're watching all that marine snow rain down, and that must be
Yeah,
full of the stuff. But if it's getting maybe denatured by the trip,
I think that is what happens because the difference between the 4,000 meter samples that I would take and the mid-water column was quite large. The 200 to 1,000 samples all sort of cluster together, which I guess makes a lot more sense. But then between 1,000 and 4,000 or 5,000, we're seeing these huge splits in the community and we're getting different structures. And I think a lot of that has to do with the benthic things that I'm detecting, obviously giving them a different signal. But yeah, we don't really get that many mid-water species coming down in the DNA. I did, like I said before, get some deep water species up on the surface, which we attributed to larvae.
What else have you found that's like completely unexpected? Because I guess a tool like this is supposed to illuminate all these rarities, like, you know, more so than we're ever going to see on the cameras.
Yeah, I think every time is a bit of a surprise. I know that's cliche and a bit boring of an answer, but I think I really like the gelatinous and the squiddies and all those sorts of lighty up pretty things. So I always find those in the samples. And I think because we don't see those as often on the cameras, I find that quite interesting and rewarding because, you know, unless they happen to go past our cameras, we probably won't see it.
Yeah.
So the giant squid was very interesting. Also the Cuvier's beaked whale, and I've got a lot of quite rare whales that we don't see very often. And then especially with the Cuvier's beaked whale, I was getting that at quite a lot deeper than its max depth was recorded. So that's something that we could look into and be like, okay, well, we're actually diving deeper than we think. And is it correlating with some of the scratches that we're seeing on the bottom of the ocean? Is that from them playing around in the sand?
That's genuinely things we've seen. We've seen them in Nova Cairn at 3,000 metres, and we've seen the whale pits at, I think, also 3,000 metres in the North Australia Basin. So I think we had a conversation about this once that we can't, there's a bit of a so what factor to that. But at the same time, no one's ever seen Cuvier's beaked whale do it. They've only seen them come back on the surface over 3,000 metres of water with mud on their face. Apparently. That's the
And I
core.
think
Could be some of that
that's-
pelagic mud.
Yeah.
Yeah.
I think that's one of the good things about how we're doing it is pairing the eDNA with the conventional or the cameras and all those sorts of things, because it's hard for me as a genomics or a molecular ecologist to say, oh, well, it obviously is deeper than we think. Because people just say, well, that's DNA. You don't know for sure. It's probably sunk down. How would you know? I think we get a lot of flack sometimes for some of the conclusions that we might draw, which is fair because we don't actually see any of these processes happening. But then pairing it with the video and being like, well, no, because they saw these big pits on the floor. It makes sense.
Yeah, there's nothing else at depth that could make a pit that size because there are no big animals there, right? So it has to be a whale or something or whatever. But
Or
yeah,
Megalodon, yeah.
yeah, hopefully. We kind of mentioned a little bit machine learning and AI, but what are the next big things? What's the next big advancements in technology?
I guess on the genetics side, I really want to look at population structures with the eDNA and can we actually use some of these sequences, and hopefully if we get them back in, you know, the best preserved quality that we can, then we might be able to look at population health and structure using genetics and some conservation genomics, and then connectivity. But then I think advancing our methods towards maybe a taxonomy-free output is very beneficial. And then I think also, yeah, like we said before, the bioinformatics, machine learning and AI is going to be absolutely huge for this because we're just producing too much data. It's too much data for just a few people. We need to be able to train the data to plug it in and be like, okay, well, you're now in the Pacific. These sequences have never been seen here before. These ones don't make sense. These are always there, those sorts of things.
I really like the idea of the long-term stuff. So recently you've bought these in situ samplers that can take multiple samples over a long period of time. And we've got obviously the big long-term observatories off here. And what I'd really like to do at some point, whenever I get 5 minutes at least to think about this in more detail, but is to do something whereby we have a long-term lander down for, you know, periodically over years and take an eDNA sample maybe, I don't know, what, every 2 weeks or something like that. Because I still think there's a lot more migration that goes on, or at least we don't know if there's any migration going on or not. At depth. And I think eDNA would be a far more appropriate way to address or at least investigate deep sea migration than cameras, because you see so little in the camera, especially long-term ones when you take a photograph like every 12 hours or something like that. You know, the chances are there's going to be nothing in it, but it would be really fascinating to see how that community changes over time. Because I still think there's a weird thing, and when I was discussing this with someone the other day about seasonality and how if these spring blooms and in the high latitudes all come down and there's this big pulse and all the rest of it, but there's also big, massive chunks, particularly in the Pacific, where they're sitting under oligotrophic gyres and nothing happens. But there's still stuff there. Now, why would a bunch of deep sea animals go to a place where there's no food all year round and there's no seasonality? Or the alternative to that is there's big migrations going on. And so the only way to answer, I think, would be long-term eDNA monitoring. I think that'd be pretty cool.
Yeah, a lot of people are starting up long-term monitoring programs, I think it depends on how much you can actually invest in these programs and what you want out of them. Because one of the benefits of eDNA is that you can get a whole ecosystem or like a tree of life view. You don't have to just target fish and animalia. You can look at bacteria. You can look at everything from the smallest of the small to the biggest of the big and target different groups. Once we get those samples back, you could use 25 primers if you wanted to amplify different groups of taxa, and then try to look at that as a whole ecosystem view. And then, like you said, seeing those changes over time would be super beneficial.
It would also work for invasives as well, wouldn't it?
Yeah, so that's where it really started. eDNA 10 or 15 years ago now was looking at invasive frogs and invasive things in river systems mainly.
Yeah.
But it is heavily used, especially over on the east coast of Australia here. They have a lot of invasive monitoring using eDNA.
It monitors human health as well. Our sewage water is routinely tested, and they charted things like the spread of COVID by using eDNA on people's poop, essentially. And it's interesting that as well that's sort of a little bit non-consensual. You know, we flush it and then it stops being our possessions, but I can't think of anything more personal than your DNA.
You gave it up. You should have kept it if you wanted it. Sidebar, but that's my old supervisor, actually. He left my lab to go and do that over in New Zealand. And we were like, that's a bit of a left turn from fish. But yeah, he started up that COVID program that you guys have.
Excellent. Anything else, Tom, you want to ask?
What do you use when you're running an analysis?
I do everything on a supercomputer. So, for example, the Trans-Pacific data that I got back, it was, I think, 400 gigabytes of data that if I tried to blast that on my own personal computer, it would, I think, immediately fry and die. So we use Pawsey here in Western Australia, and then I'll upload that, write a script, or run it using one of our pipelines at UWA Oceanomics. And then when the data comes out on the other side, it'll be a nice Excel table that then I'll go through, and then my computer will work.
Another room has gotten very hot somewhere.
Yeah, but it's not my room, so it's fine.
That's good.
It's a supercomputer one where they've got all the fancy cooling systems.
There's an ominous cube here. Can we sometimes get time on? Niwa has a cube.
They're always big black cubes. I don't know why. I think it is to make it look more ominous.
It's because it's alien technology.
Yeah.
What's the best party you've ever been to?
The best party I've ever been to. Best party I've ever been to.
An important question.
That's a huge question.
I know.
You can't just throw that at someone. They need time to think about that.
You need at least 2 weeks notice before a question as hard hitting as that.
I threw one when I finished my PhD, and I think that was just purely because of how drunk I got that I thought that was great. I don't know if it actually was a good party.
That counts. No,
Didn't
that
bother
counts.
me. Yeah. So it doesn't matter if anyone else is having a good time, because I am.
I did appreciate your honesty when I texted you to say, do you want to be a guest? And you were like, it's Friday night and I have 2 glasses of wine down, why not?
I was like, this might change come Monday, but now, sure.
But then she stood us up. She stood us up because it was a drinking night. It was a bank holiday.
That's right, yeah.
Yeah.
Weekend.
Yeah, you're like, what about then? I was like, well, I'll be day drinking, so no.
Twice it's had an impact on the podcast record. This could have been last month.
A better idea would be if we can get the microphones and record equipment and go remote and actually go track Georgia down on a bar somewhere and just do it wherever she is, whatever she's doing.
Oh, then I'd have lots to say.
Yeah.
Then I'd tell you all of the downfalls of it.
Lots of opinions.
Yeah, tell me about supercomputers right now. Well, with that, I'll say thank you very much, Georgia, for insights into eDNA.
Thank you for having me.
I did just want to follow up. We're talking about DNA homeopathy. I had an interesting little encounter, a little bit of pseudoscience. I was picking up some melatonin on the way back. I can only get it when sort of traveling. It's not available in New Zealand. So I got some of that. And the clerk at the store said, this one's actually much better value. And it was the same. It was like both were saying 10 milligrams of melatonin, but one was like half the price. And then quite subtly, I noticed it said homeopathic. I was like, well, why is there a measurement on this if it's homeopathic? And having a careful read at the back, it's 10 milligrams of homeopathic melatonin, and 10 milligrams of nothing is still nothing. But it was like really carefully, like they obviously hadn't broken any laws, but they'd really, really hidden the fact that actually it was 10 milligrams of a sugar pill with nothing in it. Yeah, I found that weirdly annoying.
But did you
I didn't
buy it
like
though?
that sort of
Did you buy
misleading.
it?
I was close. I got a picture. I've got a picture of it next to the thing I did end up buying, which had good measurable drugs.
Yeah. Want the real raw stuff. With big names you can't pronounce.
Or at least a statistically testable effect. So how are you finding dabbling into the world of eDNA? When it comes to sort of interpreting the data, when it comes out the other end?
I really like it. Actually, there's some massive potential in it. And at the moment, it's still at the stage of trying to work out what everything is. You know, you start kind of bit that library. And it's frustrating because a lot of our most abundant species or most encountered species don't necessarily have have metadata already or genetic data to blast the new stuff again. So you do have to make a little bit of inference of, is that what we're seeing on the cameras, whatever. But the thing I do like is getting a real good, interesting snapshot of what we're not seeing, because everything has bias, right? So there's a whole bunch of species we see on landers. You drive around in the sub for 4 or 5 hours, you will not see a single one of them, right? The amount of fish I come across on the sub is pretty low. It's maybe one every 2 or 3 dives. And quite often they're tripod fish that you never see at the lander and so on. So it's nice to sort of bridge that and expand that out into other ones. But what I really like is the questions you can ask about the cryptics. We are really struggling with things like Basilozetus, where you see it all the time and you know there's more than one, because there's ones that just don't look right and there's ones that are white for some reason. And, you know, there's only so many hand-wavy things you can write about how many species we're actually looking at here without catching them all, and they're notoriously difficult to catch unless you start trawling. But then we operate at 6,500, 7,000 meters, so we can't trawl at those depths. So what the eDNA would do is at least give you some confidence that it says we don't have a sequence for any of these species, but we know from the eDNA, for example, there are 5 Cuscales in that area. And then you go, okay, so 5 is a reasonable number, right? As opposed to if we came back and said it was 25, then we were like, well, there's no point even continuing this because we're so far off. But at least you get some confidence that, or if we come back and say there's only one Cuscale gene in that whole area.
You know, you can be confident that, no, they're just really varied.
Yeah, so it's another really cool tool to throw into it.
Do you think it's at that stage yet? It was the first time I'd come across taxonomy-free metabarcoding, which gave me the fears, but I was really curious to hear, like, what's that like to interpret that? Okay, that's the method. What is it like when then you are pairing that up with visuals and other methods and interpreting it?
That's where we're at. So I mean, it's not one of these new methods that supersedes something else. I think it's another line of inquiry. At the moment, the biggest problem is the deeper you go, the less there is to it. So when you get these returns and say, we think we've seen X, Y, and Z, you look at some of these like, no, you haven't. That's just the closest thing to it in the database. So it is a long, laborious kind of quality control on that, so you don't make ridiculous conclusions to it. But I think it's brilliant. I really do. And it's very easy for us to do it as well. The biggest problem we've got, and this is the reason why Georgia's aging 10 years for every human year, is the amount of data it produces is phenomenal. And so now we're at the point where, okay, we can produce huge quantities of data very, very quickly, but then you need someone like a bioinformatician to come in and start interrogating it. Or just get more people in that spreadsheet. Less people on the boat, more people in the spreadsheet.
And it must be multi-stepped. Like, you'll have a statistician and somebody to actually wrangle this data, but then you still need the sniff test of like, yeah, but that doesn't make sense. So then it's going to go back.
Yeah. And this is exactly why the centre on which I direct has people from every possible discipline, you know, and it's to have these conversations. So it's not just a bunch of ecologists or a bunch of biologists or a bunch of geneticists, or, you know, we have 1 or 2 of everything. And that's to do the whole sniff test. When someone puts it down and says they're very, very good at producing environmental eDNA, but then you also have to go, is there any way that this fish was here? And you're looking at it going, it was a goldfish. So no, that's, you know, or whatever it may be. So it's that's where I'm quite excited by the whole thing. I think it's going to be really cool.
Yeah, it's an extra thread, because ultimately we want to know what's there and what's going on. I don't like, obviously we get a lot of it at the museum. I don't like this like battle between different wings. Like everyone has the same goal. When they invented X-rays, or when we got access to X-rays, the dissection crowd didn't get all like, oh, well, that's, you know, bullshit. You can't tell unless you pull the thing apart yourself. And then we got micro CT, and it's all just more tools. We're all after the same questions. So the more data we can get.
Yeah, I think that's exactly it. It's yeah. I think there was a big argument about 20 years ago with, I read some papers, are we going to lose our sense of natural history if we keep going down the molecular route? And it was like, it was never supposed to replace taxonomy. It was never supposed to replace filming an animal in situ, right? It's just that there's a lot of things we can do by filming them alive. There's lots of things we can do by examining them dead. And there's now lots of things we can do genetically. And eDNA is like just an addition to that. Now we can detect where they are without even seeing it or sampling it. See what I mean? So it's just this, it enhances everything else.
It's helping you interpret the visual.
Yeah.
And you still need the sniff test. Like no one went and executed all the experts. That you still need the sort of, but does this make sense?
If you think about things like the big fin squid and that whole thing we're talking through with like Vecchione, is the range, the depth and the geographical range of an animal like that is only very, very slowly being teased apart because you're waiting for someone to come across it by mistake. Right, and then you go, okay, so it goes to this latitude. Okay, you now see it off Tonga or wherever it may be. Okay, it goes a bit deeper because someone's just saw another one, and that process could have taken 10 years to make those little nudges as to where this animal is. Now, theoretically, you don't need to disturb it. You don't need to go looking for it. You don't have to wait for it to happen. Just sequence it in eDNA and then say, okay, the big fin squid lives all the way to Antarctica. Maybe. It answers that question much more rapidly. But there are also questions about why the hell does it have such bizarre arms? That's not going to be answered by eDNA. It won't be answered by genetic information and molecular stuff. It probably won't necessarily be resolved entirely by having a dead specimen on the bench.
It'll be all those bits coming together.
Yeah. If you just film it, and hopefully at some point somebody, go, okay, that's what it's doing. And then so there you go. So it just depends what the question is. It really does.
There's weird glands on the elbows. Nobody talks about that.
Yeah. Yeah.
There's elbow glands.
What do they do?
Can I offer a pet theory?
Yeah.
I think they're buoyant. I think they're filled with ammonia or something to make the elbow a little bit buoyant so they don't have to hold up all that massive arm.
Oh, I see. Yeah. Yeah.
It's got a little floaty. It's got a little armband.
Oh, yeah, that makes sense.
Yeah, I'm up for that one.
Yeah.
I said that to a squid person and their eyebrows went up in a way of not outright laughing at me.
Like, one, that's stupid, and two, stay in your lane.
Yeah. It makes sense
We'll
to
make
me.
the ridiculous hand-wavy statements about squid, not you.
I wanted to talk about a little project we've been involved with as well as we move into this area. And actually, I know some museums are a little bit sort of feel at war with genetic work, but we've sort of really embraced it. And actually, most of our loans now are genetic and tissue-based rather than sending an actual specimen, because this is a big area of research. One big project we've been involved with is to have a really good database of at least New Zealand's fauna. And part of the advantage to doing it with that is there's a lot of stuff on the big databases which the specimen is gone. And if there's some doubt about its ID, or even worse, that species has changed since, then a lot of these databases, if you don't have a voucher specimen tied to them, you can't go back and look again, if that makes sense. So like if one species gets split into two and you go onto GenBank, well, which one do I have? And we threw it out. So we can't go back. And you'll be able to tease it apart, maybe genetically digging back into it, but you've lost that little bit of confidence. So you almost need the genetic holotype. You need the reference, you need the voucher to underpin those sequences so you can go back and check if things change. It's quite exciting stuff. No, it's just another tool. I would not refuse to use a CT scanner because I like dissections or I like good old-fashioned X-ray. More tools. I want to know about the things. It's not teams. I'd want to know about the things. And if someone can help me learn more about the things, I'm up for learning about the things.
Yeah, I agree. Yeah, that's cool. I think it's going to be stuff that's going to come out soon. It's going to be quite incredible.
Ooh.
I mean, George has had eDNA 3 Niskin bottle samples on every land that we've deployed over the last 2 years, which comes to 485 times. Right across abyssal plains twice, all across the entire depth range of Nova Canton Trench or Trough, from 800 metres to 10,800 metres in the Tonga Trench, and right across the South Shetland Trench in Antarctica, and over the Falklands Escarpment. That's how much data we're sitting on right now. It's like, ooh, super cool.
Very
That's
cool.
a 10,000 metre sampling range in one place. And we have got a lander at least every 90 metres, over 10,000.
I do find it interesting how we're all having our poops and peas monitored, and things like COVID outbreaks are being tracked by eDNA of the sewage outlets, which it came up in it a little bit. And I know Georgia was joking when she said like, oh, but you threw it away. I just find it interesting, a little area for like privacy and consent. Like just flushing your poop doesn't consent to have your genome sequenced.
Are they actually sequencing your genome from that?
I
Or
don't-
Are they not just looking for general popular? How do they pick you out from anyone else?
Well, same as how you're picking up fish from other fish.
I know, but they'll never know it's you. They could have your genome, but they wouldn't be able to track it back to you because it came out of the sewage pipe with everyone else.
This is true. But
So
it's still,
it's not a security breach. The feds are not going to be coming around and going, "You're the guy."
you feel that the toilet successfully anonymizes the participants.
Yeah.
I don't know, I feel weird about it. I had a chat with my mum when she was sort of encouraged by a family member to do the 23andMe. I don't know why it makes me so nervous, but I was like, that's not something you can kind of take back. And yes, they've got a policy, but what could people do with that in the future when it's such an emergent field? And now that's kind of come to pass because they've gone bankrupt. And so all the agreements that you signed, it's now a company that's being stripped for parts, and somebody might be able to get that data set, and and you didn't sign an agreement with them about how they could use it. So yeah, I don't know. It's just such an emerging field, and I get a bit worried about it.
Well, on that note, just keep worrying, Tom.
Just keep worrying. Just keep worrying. Just keep worrying.
Is there any way you can connect your worry to your phone and see if you can charge it?
Oh, I'd blow up. You know, I'm powered by worry.
Yeah, so I mean, just rather than it just being a waste of energy, just charge something of it, like a contactless charging station on the top of your head, and that your worry is the bit that powers it, and you just put the phone on top of your head and charged. Done.
I'm thinking more sort of like creepy X-Man thing, like
just gives them a lifetime of worry in a split second.
Oh yeah. Oh, you can just give people panic attacks. That's quite a good X-Man power, actually.
You start weaponizing anxiety.
Yeah. Oh, that's it. That's it.
Yeah, I'm gonna get a lifetime of anxiety in 1 second. That'll knock you sideways, wouldn't it?
What's the X-Men character name? Is it just Mr. Anxious?
Yeah. Thanks, man.
Angstman. The Angstman comer. And that concludes this episode of the Deep Sea Podcast. Check out the show notes for a list of everything we've spoken about and some links for some wider reading. Also links to our merch and our Patreon. And we really can't thank the people who join our Patreon enough. It is what allowed us to make this self-sustaining. As a little bit of a reward for that, you do get access to the Discord, which is nicely developing as a really nice little community, really. People are getting to know each other really well. We've got a few scientists on there, but also regulars on the show and just really passionate listeners. And actually, it's a really good space for discussion. A lot of the news gets discussed and broken down on there. And I'm finding it really interesting to see other people's take on it. So actually, the show is getting stronger because of that community, and they're very much a part of it. They certainly influence what we choose to talk about and how we choose to talk about it. So really big thank you to those. So until the next time, unless I get eaten by a shark, it's funny now, but it won't be if I really do get attacked by a shark, which I might. But anyway, until the next time, which I'm definitely going to be alive for, we'll deep see you next time, and we abyss you already. You're still wearing the socks from the airplane.
Oh, absolutely.
They don't do that anymore. Was it Virgin Atlantic that used to give you the brightly coloured
Oh, you
socks?
still get socks on airplanes. You just got to do business class. You get all sorts of things.
Oh, right. No. Yeah. It's been a while.
Yeah. All sorts of
what else
interesting
do you get?
trinkets now.
Champagne and the big proper headphones.
Yeah. And an iPad and a
What?
screen. Yeah. Well,
An iPad and a
the
screen?
airlines in the class in which I normally travel, Tom, you get all sorts of this. You know, if you don't want to sit in the same position watching your massive, big, huge HD TV screen, you can roll over in your bed and watch something on an iPad.
Wow.
Yeah.
In spoiled toddler class.
Yeah, it's a tough
in
life.
In a little cot.
Yeah.
I'd give you a hard time if I didn't know how hard you do work on either end of that. So you've earned it, mate. You can have a little cot. You can have an extra iPad. Like, extra iPad is a treat because it's the
Yeah, the
weekend.
best thing about traveling business class all the time is everyone who does it is insufferable, and it's actually quite funny just to see how much you can push it. Oh, dear. I don't know
Because
why.
I have travelled with you, they probably won't have appreciated that.
No,
You still wear a big wax jacket for the entire
No.
flight.
but yeah, not
That was the good old days. Sweating out the beers and in a wax jacket.
I was telling someone else about this recently about that flight we did to Puerto Rico, and as far as I remember, is I fell asleep on the plane, got up at the end, and we went into San Juan, and then you just sort of fessed up that you had some sort of mad seizure and migraines and all sorts of stuff during the whole entire. You were sat next to me. I didn't notice once that you were actually requiring and receiving medical attention for most of the flight. And I was like, oh well, take me get off. Tom.
And off. Yep. I envied you.
Yeah, I had
Yeah,
no
that
recollection
one
that
was rough.
whatsoever.
Don't know what triggered that, but yeah, I was just just sweating and writhing like a drug addict coming off.
Puerto Rico.
Good times.